Okay, let's do the protease. So I've explained the structural conventions. That's the protease structure up there. And this is called the bulldog conformation. You can see the sort of floppy ears that sit down. There you go. Now the bulldog sideways there. It's like the head of a dog. Yeah. With it actually has the substrate in the center here. That's this sort of molecule that you can see running through the middle. So that's where the protein actually fits that's going to be cleaved, and these are going to lock on around it. As you can see, the protease is dimeric, which means there's two of them. So you get the green copy and the orange copy that together are going to work and both cut substrates. The active site, if we pause this for a minute, is going to be on this loop. It's right about here. And that's going to be an aspartic acid, which is amino acid D. So that's going to show up as a purple column in our alignment. All right, so spin away, little bulldog, and let's go look at the alignment. And so the alignment is in gel view once again, which you're welcome to download and play with this as much as you like. So this is the whole Pol region with the protease all the way through the end of the integrase from HIV-1. These are only HIV-1s that are in the alignment because some of these parts don't align as well. The protease rather gets a little ropey, and the tail end of the integrase gets a little ropey in the multiple sequence alignments. They're just less conserved. Protease is little, so the actual conserved part's going to start right around here, around 70 in this alignment. And the active site is going to be this sort of E-A-L-L, and it's the D right after that. That's the aspartic acid. That's the actual one that's going to do the catalytic work here. You can see some of the rest of the protein, and it's going to go down to around this point, right where it starts to turn sort of basic. That's where we're into the reverse transcriptase. So the cleavage is going to be in this neighborhood. The other important features that you want to see are going to be right here, around 120 in the alignment. And this numbering is based on the alignment. And remember, the colors show conservation. So amino acids will only be colored if they are conserved, if they appear over and over and over again in multiple copies of HIV, which for a virus that changes as much as HIV means that these are things that are very hard to change. Rather, they're easy to change, but when you change them, it's hard to be a functional HIV anymore. They're basically lethal mutation zones where most changes are not well tolerated. All right. And so the things you want to look for here are going to be the substrate binding pocket, and that's going to be these guys right here, sort of flanking this GGI-GG region, and the little amino acids either side of that, which are blue, so those are going to be hydrophobics for the most part, and it's going to be framed by these little basic lysines for the most part, either side, which are the little red columns that you've got over here. And so there you go. That is the active site. These glycines are going to form the oxyanion hole, which is important for stabilizing the intermediate step. So this is going to work like other proteases. It's going to make an unstable intermediate where you've got a carbon that's going to be trying to hold on to 5 things instead of the 4 bonds that carbon would like to make. That thing is so unstable that you can tell it's there from the energy that's released, but you can't really see it there. And so this is an energetically unfavorable thing that happens in the enzyme as part of the mechanism. That's why the enzyme is an enzyme, because it allows for this improbable thing to happen reproducibly. Yeah. So there you go. That's an enzyme. Looking at the enzyme structure itself, you can see it's basically a beta barrel on either side, and you've got kind of the start of another beta barrel over the top, these sort of loops that are going to reach out and join each other. Mostly beta stranded. There's one little helix down here kind of capping off the bottom of the barrel, but that's not where the important stuff happens. The important stuff all happens in this sort of void at the center. And there you go. That is your HIV-1 protease. the active site over here, and your substrate binding pocket, including the oxyanion hole over here. All right. Now you've at least got a little more of a picture for this one. Thanks very much.